Welcome to the Anatomy and Physiology section in Chapter 8 for muscle metabolism. We're going to look at energy for metabolic reactions. As we've already learned previously, energy is required for all cells to do work, and we know that energy can come from a variety of different sources as far as the amount of heat, light, sound, mechanical, or in this case, chemical energy that we're going to be working with in muscle and understanding muscle contraction. Energy is involved in all metabolic reactions, both anabolism, which is the building up of large molecules, and catabolism, which is the reduction of larger molecules into smaller ones. The chemical energy that you're most familiar with that we're going to be working with muscles is in the form of ATP. ATP, or adenosine triphosphate, is a molecule that is able to store energy in that high-energy phosphates and allows molecules to do work. Cellular respiration is the process used to produce those ATP molecules. Cell respiration occurs in 3 steps: glycolysis, the citric acid cycle, and the electron transport chain. Producing carbon dioxide, water, and that ATP, which is a form of chemical energy, also produces a large amount of heat that is generated in our body. We know that oxygen is required for this process, both at the beginning and end of cell respiration, and without oxygen, we produce very little ATP, typically only 4 molecules per molecule of glucose, whereas aerobic reactions that do have ample sources of ATP produce the most ATP available. The first step in cellular respiration is a process called glycolysis, and just like its namesake, using glycogen as a model, lysis means to split, so we're going to break down a molecule of glucose from a 6-carbon molecule into 2 pyruvic acids, which are composed of 2 3-carbon molecules. This occurs in the cytoplasm. Notice it's stated here this is the anaerobic phase. It does not require any oxygen present for the glycolysis to occur. We'll produce 2 ATP molecules per molecule of glucose. As you'll notice below, it says summarized by 3 main events. One thing we do know is that the phosphorylation does require 2 ATP to be formed. Here. After the phosphorylation of glucose, that's split into 2 3-carbon pyruvate molecules. Along with the pyruvate molecules, the 2 of them that are formed, this process also produces 4 NADH molecules, in addition 4 ATP. The NADH molecules are reduced in that fact and will be used later in the electron transport chain. We can see here from this slide on glycolysis, the first step is to again take the 6-carbon glucose molecule here and phosphorylate it with those high-energy phosphates. That does require 2 ATP, referred to as the priming phase. Immediately that molecule is fractured into 2 3-carbon molecules, what we call splitting or cleavage. Okay. These 2 3-carbon molecules are then further reduced later in the next step. For event 3, for the stage of glycolysis, we'll notice we take the 2 3-carbon molecules formed from the cleavage of glucose, and we form 4 additional molecules of ATP. Those are phosphorylated. In addition, NAD is reduced to NADH. The NADH molecules will be used later in a step called electron transport chain. As we see in the bottom of the diagram, if oxygen is present, those 2 pyruvic acid molecules will enter the citric acid cycle, forming what we call acetyl-CoA, and also producing 2 molecules of carbon dioxide as a waste product. Notice on this slide, if oxygen is not available, the 2 pyruvic acid molecules, or what we refer to as pyruvate, do not enter the citric acid cycle and Krebs cycle. They immediately are converted into lactic acid. As we know, this product can build up in the surrounding the mitochondria and changing the relative pH of that environment. In addition, ATP production is much less, acquiring only the 4 ATP that we end with. If oxygen is available, we move into what's called the aerobic reactions, aerobic meaning involving oxygen, first being the Krebs cycle. Those two pyruvic acid molecules enter the mitochondria from the cell cytoplasm and immediately enter a cycle where the acetyl-CoA is formed by cleaving another carbon, forming a molecule of carbon dioxide, waste, immediately binds to the namesake of this process called citric acid, forming again a 6-carbon molecule. And immediately following that, 2 more carbon molecules are cleaved, forming 2 ATP due to phosphorylation. And as you can see, that cycle also produces some more of the NADH and FADH2 molecules that are reduced with those high energy. The last stage in forming a number of ATP molecules from our glucose is called the electron transport chain. And just as it states here, if you notice from the diagram, the membrane lining the mitochondria consists of a series of folds and presents an area where ions can move across this membrane in a process called chemiosmosis. This process is where we're going to form the majority of these ATP molecules. The NADH and FADH2 molecules, these high-energy electron carriers that were formed earlier in this process, here will be oxidized and passing on that electron ultimately to a molecule of oxygen, forming water in the process. The last stage in this aerobic reaction process is called the electron transport chain. This is where we see a number of those molecules that are produced earlier in this process come into play. We also note that conversion of these molecules requires a number of enzymes, first being cytochrome oxidase. This is the enzyme that helps to transfer those high-energy electrons to oxygen, forming water molecules. We also see the role of NADH, dehydrogenase. This enzyme removes hydrogen, basically oxidizing NADH and providing that high-energy electron across the movement across the membrane. And lastly, ATP synthase. ATP synthase uses the membrane found inside of the mitochondria to shuttle these high-energy electrons across that membrane. In doing so, provides an electrochemical gradient. And that electrochemical gradient is the process where we help to form ATP. Here we see within the folds of the mitochondrial membrane the NADH dehydrogenase removing or oxidizing the NADH, providing a high-energy electron carrier. The passage of that electron carrier across the membrane allows the final enzyme ATP synthase to form a molecule of ATP. The last stage in electron transport chain forms a total of 38 molecules of ATP. If you remember, in the beginning of glycolysis, we require 2 ATP molecules to phosphorylate that glucose, to cleave that into a 3-carbon molecule, so we're reduced to a total of 36 ATP. Each ATP molecule produced is comprised of 3 parts: an adenosine molecule, a ribose molecule, and 3 phosphate molecules. Now these phosphate molecules occur in a chain. As you can see down here, the third being a high-energy bond, a high-energy covalent bond. And we know covalent bonds do retain a lot of energy, and when that bond is broken, we release that energy and can use it to drive metabolism. That leaves us with a molecule of ADP. ADP can also be phosphorylated using the enzyme creatine phosphokinase, along with energy captured from cellular respiration, converting our food into energy, and resynthesizing that molecule of ATP. Creatine can be found as a supplement, oftentimes in many foods found in health food stores, but typically our liver can synthesize ample amounts of creatine to promote the formation or donation of that hydrogen phosphate to ATP molecule.